Welcome to the Lymphoma Research Foundation's update on CAR T-cell therapy. I am Jimena, and I'll be the operator for today's call. During today's call, you will hear from an expert speaker, and you will have an opportunity to ask questions. If you have questions during today's presentation, you can ask them at any time in the Q&A box of the webinar. As a reminder, this webinar is being recorded. At the end of the program, a link will appear on your screen. Please follow this link to complete the evaluation of this program. If you are listening on the phone, this link will be sent to you via email at the end of the webinar. And now I am pleased to introduce Jessie Brown. Jessie Brown is the Associate Director of Patient Education at the Lymphoma Research Foundation. Welcome, Jessie.
Thank you, and thank you to each of you for taking the time to join us on today's update on CAR T-cell therapy webinar. Before we begin, we'd just like to thank our sponsors of this webinar, Genentech, Kite, a Gilead company, and Novartis. Before we begin with the program, I'd like to share some information about the Lymphoma Research Foundation. We're the nation's largest nonprofit organization that focuses specifically on lymphoma, and our mission is to eradicate lymphoma and serve those touched by the disease. All of our work is led by our Scientific Advisory Board, which is a group of 45 leading lymphoma experts from around the country. In addition to today's program, we have a variety of other programs and services available to you. One example is our LRF helpline, which serves to complement our patient education programs. The helpline has master's level trained staff who provide individualized information on all types of lymphomas, as well as information on treatment options, clinical trial navigation, and connections to resources such as financial assistance. We offer a peer support program called the Lymphoma Support Network. Through the Lymphoma Support Network, we can connect patients and caregivers with others who have been through similar experiences for emotional support. In addition, LRF has free comprehensive disease guides and fact sheets, which can be ordered or downloaded on our website, lymphoma.org. We also offer a variety of other in-person and virtual educational programming, so you can continue learning about the latest updates through the year. And finally, we have an award-winning mobile app called Focus on Lymphoma. The app provides helpful disease content as well as unique tools to help you better manage your lymphoma. We have a wonderful program planned for you today, and I'm honored to introduce you to our speaker, Dr. Manali Kamdar. Dr. Kamdar is a hematologist and oncologist at UC Health University of Colorado Hospital, where she is the clinical director of lymphoma services. She is also an associate professor of medicine at the University of Colorado Anschutz Medical Campus. Thank you, Dr. Kamdar, for speaking at our program today, and I'll now turn the talk over to you.
Thank you very much, Jessie, for this introduction, and good morning, good evening to everyone. Here we're going to talk about all the updates on one of the most promising therapies of the decade, which is CAR T-cell therapy. And CAR T-cell therapy has left quite a mark in most of the heme malignancies, particularly lymphoma. So my talk today is going to focus on CAR T-cell therapies in patients with lymphomas. So for the longest time, probably about 3 to 4 decades, in terms of the toolkit that we had to treat lymphomas, we typically had the age-old concepts of killing it all, which is not just the cancer cells, but also at the cost of normal healthy cells. And these tools used to be chemotherapy, radiation treatments. As a result, patients, of course, derived benefit, but it ended up at the cost of killing some of the normal fast-growing cells. In the most recent contemporaneous era, the tools for treating lymphomas have radically changed. And one of the most recent concepts has been immunotherapy. Of course, it made it to being one of the most significant advances of the decade. But the idea behind chemotherapy is to really only target cancer cells and utilizing the patient's own immune system. As a result, we typically would spare the normal healthy cell, thus hoping to derive efficacy, which is cancer kill, but not at the cost of increased toxicity. In terms of moving forward with what is immunotherapy and what does it mean to utilize your own immune cells, I think just as a way of background, it's important to recognize that all of us are born with 2 distinct types of immunity. And this immunity typically kicks in whenever we are exposed to a foreign antigen. This foreign antigen could be just a germ, it could be a cancer cell, and it typically triggers two types of immunities based on the timing of exposure. For example, if someone is typically affected with a cancer antigen, for the initial 12 hours, something called innate immunity kicks in. Innate immunity is typically made up of cells such as dendritic cells, phagocytes, NK cells, and it is able to ward off that cancer antigen. Right after 12 hours sets in something called the adaptive immunity. The adaptive immunity typically consists of T lymphocytes and B lymphocytes. And as a result, it typically has the ability to fight it off for a much longer period of time because usually the adaptive immunity has memory, which means it can then recognize that cancer antigen as a foreigner or an enemy for the rest of their lives. Now, clearly, in patients who develop lymphomas or any other cancer, there is a dysfunction that happens at the immune level, which means for some reason, even though the T lymphocytes are there that can typically recognize a foreign antigen as an enemy, it's not able to do its function right. As a result, it could end up being exhausted. Versus sometimes it just loses the memory. And therefore, this is a unique way in terms of treatments for targeting lymphoma management, where we can typically manipulate patients' own immune cells to make them fighter cells again. Basically, a memory that they no longer have. We hope to induce that memory with agents that can reactivate the immune cells. So that's the basic concept of immunotherapy. And as you may imagine, that has been ongoing for nearly 4 decades now. Previously, some of the immune treatments that have been currently also utilized is, say, for example, antibody. A lot of lymphoma patients, especially B-cell lymphoma patients, get rituximab. Rituximab is an antibody that targets an antigen called CD20 on the lymphoma cell. And as a result, you can typically target this antibody against the lymphoma cell, thus inducing lymphoma cell kill. Rituximab has been around for nearly 4 decades. Similarly, allogeneic transplants has been one of the immunotherapeutic toolkits that we have used for several decades. Now, since then, we have made a lot of progress, but we are beginning to see the fruits of the labor that have been ongoing for nearly 3 decades. And I would say one of the sweetest fruits of all here has been the implementation of CAR T cell therapy. So in terms of the immunotherapy landscape, as you may imagine, the center of this cell is the lymphoma cell. The lymphoma cell expresses certain antigens or proteins that you can technically target by either making agents that can sit on that target and then kill the lymphoma cell, or it can typically kill it by inducing your own immune cells, such as T cells, to kill the lymphoma cell. And there are several different immunotherapy options that are currently available, that are FDA approved, as well as many others that are currently in clinical trials. But like I said, the focus of today's talk is going to be on CAR T cell therapy. And this CAR T cell therapy has certainly done a whole lot of good for a lot of patients who previously did not have options beyond just chemotherapy. So this is just a quick refresher on lymphomas, because as I can imagine, there might be patients with several different types of lymphomas. Lymphoma is not one disease. It's actually made up of 108 different subtypes. Now, of course, I'm not going to bore you with individual types, but primarily it's divided into either Hodgkin lymphoma or non-Hodgkin lymphoma. Non-Hodgkin lymphoma is way more common than Hodgkin lymphoma. Nearly 80,000 patients in the United States are diagnosed with non-Hodgkin lymphoma every year. Non-Hodgkin lymphoma is further divided based on subtypes called B-cell non-Hodgkin lymphoma or T-cell non-Hodgkin lymphoma. I am going to focus today on the more common non-Hodgkin lymphoma, which is B-cell non-Hodgkin lymphoma. It makes up for nearly 85% of all non-Hodgkin lymphomas. Now, B-cell non-Hodgkin lymphomas are further divided into 45 different subtypes. And typically, I like it simple. And in terms of assessing which subtype of B-cell non-Hodgkin lymphoma a patient has, it's further divided into 3 buckets. One can either have an aggressive lymphoma, which is very fast growing. Typically, chemotherapy is the first line of tools that we use in taking care of patients with aggressive lymphomas. Albeit curable in about 2/3 of the patients, I would say 40% of patients may relapse. And those are the patients who previously didn't have a lot of good options, where CAR T cell therapy has made the biggest impact. Aggressive lymphoma, such as diffuse large cell lymphoma, primary mediastinal lymphoma, Burkitt lymphoma, these are some of the aggressive large cell lymphomas. Then we have the second bucket, which is the indolent lymphoma. Indolent lymphomas are usually non-aggressive lymphomas. These are typically slow-growing lymphomas. They typically don't come in with a lot of symptoms, and they are chronic illnesses. So we typically manage them based on where the patient is at in their life, what type of a tumor burden do they carry, what is the impact of the tumor on the patient in terms of their blood work, in terms of their symptoms, and that really helps us understand how do we best treat these patients. So it's certainly treatable, and in a lot of times, treatments will get them into remission, but these patients have a tendency to keep relapsing. So that is what we want to make sure stops happening. And that's where I will say CAR T cell therapy made a big splash as well. Then, of course, we have lymphomas, where I call them mixed lymphomas, but it typically harbors the features of both the non-aggressive lymphomas as well as the aggressive lymphomas. And one of the poster childs of mixed lymphomas is something called mantle cell lymphoma. So it can initially present as a slow-growing lymphoma, to later becoming quite aggressive and fast-growing. Previously, we only had chemotherapeutic options, but for these lymphomas, CAR T cell therapy again has made a huge impact in making sure that these outcomes for these patients have improved significantly. So let's deep dive into what is CAR T cell therapy. So CAR stands for chimeric antigen receptor. In the simplest of terms, it's basically taking patient's own blood, isolating the T cells, which are the fighter cells for that patient, taking it to the lab, and through a vector-induced genetic recombination method, we basically make them super fighter cells. Super fighter cells that are now able to recognize proteins on the lymphoma cell as a foreigner and kill it. So basically, these patients' own T cells are becoming more activated T cells called CAR T cell. And here, as you can see on my pointer here, this is the construct of the CAR T cell. It's basically made up of 3 domains. The first domain is called the antigen recognition site, which is the site that latches on to the protein on the lymphoma cell. Now you may imagine that this protein could be any. But for patients with B-cell lymphomas, the CARs that have made a huge impact are the CARs that target a protein called CD19. So this antigen recognition site for patients with lymphomas where we use CARs is against the protein CD19. Then this is the second agent, which is the transmembrane domain and the co-stimulatory molecule. Basically, the co-stimulatory molecule can be different based on the type of construct we use. It can either be CD28 or it can be 41BB. The others are in clinical trials. And the idea of the co-stimulatory molecule is to basically make sure that the signals continue to be sent to the signaling molecule and they remain persistent. And this signaling is the T cell activation. And this is the T cell activation that basically activates what the T cells are supposed to do, which is cause lymphoma kill. So, with that in mind, there have been several generations of chimeric antigen receptors or CAR T cell therapies that have been in research for decades. The first iteration was something called the first generation CAR. I think of it as a simple CAR. And as you can see, there is the antigen recognition site, there is the T cell activating domain, but there is no co-stimulatory domain. And because there is not the co-stimulatory domain, these CARs worked, but they unfortunately did not persist or expand. As a result, there were short-lasting benefits of this CAR T cell therapy. Then came the introduction of the co-stim domain, which was either CD28 or 41BB. And this, I would say, is called the second generation CAR. And in my opinion, it's like a muscle CAR. And this is where we have made the biggest impact for our patients with lymphoma. And most of the FDA constructs that we will be talking about are the second generation CARs. The third generation CAR is basically CARs that have multiple features, not just one co-stim domain, but could have two co-stim domains, could have a domain that can induce certain substances that can make the CARs more persistent. And this is like the race CAR. But the race CARs right now are in clinical trials, and I think we're just waiting to see what the efficacy of those CARs look like, if they are better than the muscle CAR, and they are better and less toxic than the muscle CAR. Because at the end of the day, we absolutely want to make sure that the quality of life for our patients is preserved. So let's talk about the initial impact that we have made for patients with CAR T cell therapy. Unfortunately, this slide is not being pulled up. I'm sorry about that. But basically, I will say that the first report for CAR T cell therapy was initially in 2010. And in 2010, this CAR T cell therapy was initially tested in patients with chronic lymphocytic leukemia. And it was done at the University of Pennsylvania, and that patient remains in a remission and is doing extremely well. Similarly, you may have heard of the success story of this child, now a woman, Emily Whitehead, who was diagnosed when she was a little girl with a lymphoma called acute lymphoblastic lymphoma. And this patient was one of the first few patients who got a CAR which was targeting CD19 called tisagenlecleucel. And she had had at least 6 to 7 lines of previous treatments. And after CAR T cell therapy, finally achieved a remission. And this is basically Emily Whitehead today. She was cancer-free here and now is cancer-free nearly 10 years later. Suffice to say that she has been cured of CAR T cell therapy of her ALL. So clearly, this is gratifying stuff, and it's important to recognize how it's all done. So CAR T cell therapy is a 3-step process. As you may imagine, we first have to take the blood from patients, and that process is called apheresis. It's typically a 1-day commitment to a cancer center's apheresis center where they collect your blood. And this blood is collected so that we can actually isolate your T cells from it. Apheresis sample that's collected is then sent off to the laboratory where the magic happens. And this is where CAR T cell therapy are made. It typically takes 3 to 4 weeks to make CAR T cells in the lab. We insert the gene for CAR into the patient's normal T cell, which has stopped recognizing lymphoma, and thus the patient has lymphoma. And through this method, we now make these T cells express the chimeric antigen receptor, which can now bind lymphoma cells and recognize them as the enemy. And this process takes 3 to 4 weeks. Now, you may imagine that for about a month, the cancer is not going to wait. So a lot of patients may get something called bridging therapy. And usually bridging therapy may be low-dose chemotherapy, that the idea is to only keep the lymphoma stable. If there is cancer kill, that's great, but The idea is to just make sure that the patient remains stable while the CARs continue to be manufactured to be then returned to the patient. Usually 4 weeks after that, we get a call from the company saying the patient's CARs are ready, and this is where the plan starts. Typically, there are 3 days where we give patients low-dose chemotherapy. This low-dose chemotherapy usually contains chemo agents like fludarabine and cyclophosphamide. And the idea behind this is not to kill tumor. The idea behind this is to basically make sure that we create an environment within the bone marrow that is now accepting of CARs so that the CARs can expand and persist. So the idea behind giving the low dose chemo is very different than how we do chemo otherwise. This low dose chemo is meant to make CARs persist and expand and thus function effectively. Once the low dose chemotherapy is given over 3 days, the side effects, by the way, are very minimal and tolerable. Day 0 is what we call it. We infuse the CARs back into the patient. That day is fairly anticlimactic. Patients don't really have a lot of symptoms then. But the idea is when the CARs go into the system, they unleash upon the cancer cells because their receptor on the T cell now starts recognizing the protein on the lymphoma cell as foreign and starts attacking it. And that's basically how the CARs start killing lymphoma cells. At this point in time, we have nearly 4 FDA-approved CAR T cell therapies, and they are all at this point targeting CD19. The 3 most common, which are approved for patients with aggressive large cell lymphoma, are axi-cel, axicabtagene ciloleucel, tisagenlecleucel, also called Tisacel, lysacabtagene maraleucel, also called Lysacel. You can see then that the difference here, besides the manufacturing process slightly, is different for Lysacel versus Tisacel and axi-cel, but the co-stim domain is what sets them apart. For axi-cel, the co-stim domain is CD28. For tisagenlecleucel and lysacabtagene, it's 41BB. And there's a slight difference in how this co-stim domains work. So with the CD28 co-stim domain, there is a rapid expansion of CARs the minute it enters the body. As a result, we do end up seeing slightly higher incidence of inflammation. And I'll go into that in a little bit more detail later. On the other hand, with 41BB molecules like Tisacel and Lysacel, they go in and expand very slowly. As a result, the side effect profile might be slightly more favorable with Lysacel, Tisacel versus axi-cel. Moving forward, wanted to give you an example of one of the early trials that we had open at my site here at the University of Colorado nearly 5.5 years ago now. This was a patient, as you can imagine, you can see that these are the 2 tumor masses. The mass in his abdomen was nearly 30 centimeters. And this patient came to me after having had 5 lines of treatment. So lots of chemotherapy, radiation, and nothing worked. He subsequently enrolled on the clinical study, and at that point I had the clinical trial open with lysacabtagene, and on the day 30 mark, the entire tumor here had completely resolved. So the patient had achieved a complete response, and the patient remains in a remission 5 to 7 years later now, and I can safely say he has been cured of his lymphoma. So similarly, there have been experiences with all the 3 constructs. And I'm showing you curves which are called the Kaplan-Meier curves. And whenever we start seeing a plateau emerge like that, we know that patients have actually stopped relapsing. This is basically excellent response for a patient cohort who was heavily pretreated with multiple agents. And that's why previously, or initially, when CAR T got approved, it was approved in the most aggressive lymphoma set for patients who had failed at least 2 lines of treatment. Meaning, if somebody had diffuse large B-cell lymphoma, they had failed R-CHOP, subsequently they had failed another line of chemotherapy, like say R-RICE or R-GEMOX, So they were heavily pretreated, the cancer burden was growing, and in those patients, we were able to establish that nearly 40% of these patients now, 5 years later, continue to be in a remission. Needless to say, they have been cured of lymphoma. So previously, based on the FDA-approved CAR T-cell therapy, there was a shift in the algorithm for patients with relapsed refractory diffuse large cell lymphoma. Meaning, for patients who relapsed, we typically gave them another round of chemo. If they responded and were transplant eligible, we took them to an auto transplant. But if they did not establish chemosensitivity, we got them to CAR T cell therapy. Similarly, if auto transplant failed, CAR T cell therapy was, of course, an option. With this strategy, 40% of patients got cured. So the obvious question is, why don't we move CAR T cell therapy forward? And with that in mind, we looked at where was auto transplant ineffective. And we found that in patients who just didn't respond to frontline chemotherapy, or in patients who relapsed within the 1 year mark of receiving R-CHOP, these were the patients who didn't quite derive a lot of benefit from auto transplant. And that's what set the biggest of the big 3 randomized trials actually targeting CAR T cell therapy to CD19 CAR T cell therapies against auto transplant. So it was really going forward to making sure auto transplant was tested against CAR T cell therapy in patients with high risk second line relapsed diffuse large B cell lymphoma, meaning they had received only 1 line of chemotherapy. So now we were testing CAR T versus transplant after patients had failed 1 line of treatment. And the net is that of the 3 studies that was set, 2 studies, meaning ZUMA-7, axi-cell, and TRANSFORM, meaning lyso-cell, these 2 studies showed a significant advantage to CAR T-cell therapy versus standard of care, which was auto-transplant. Now these curves, if you can see, there is a big split, which means the standard of care curve did not do as well as the lyso-cell curve or the axi-cell curve. And very quickly, the FDA approved lyso-cell and axi-cell for patients who had relapsed with high-risk diffuse large B-cell lymphoma after frontline chemotherapy. And thus in 2022, this was the new algorithm. Not everybody needed to get another line of chemotherapy to establish whether they're going to respond or not. So number one, we saved people 1 line more of chemo treatments, which were more toxic. And in patients who are high risk, we could typically take them now directly to CAR T cell therapy. Lyso-cell also was explored in patients who relapsed after R-CHOP or R-chemo and were not transplant eligible. And in those patients, lyso-cell showed excellent responses. So now, in patients who are transplant ineligible, CAR T cell therapy can also be offered with lyso-cell in the second line setting. So as you can see, in just a matter of 4 years, we have made significant advances for patients with aggressive large B-cell lymphoma. Moving forward, of course, there's the yin and the yang. And if we have efficacy, we want to make sure we also look at the toxicity. Now, CAR T cell therapy is not chemotherapy, it's immunotreatments, which means it activates immune cells to unleash on the tumor. And when that happens, they sometimes can cause the tumor kill, or the war that they are waging against the lymphoma cell can release inflammatory substances called cytokines. And therefore, there are 2 unique toxicities that CAR T cell therapies can cause in patients who are receiving them. One is called cytokine release syndrome. The other one is called neurological toxicity. And previously, I will confess that when I initially opened the first CAR T cell study in Colorado, we didn't quite know how to manage these toxicities effectively. But over the last 5.5 years, we have learned and we have now really been very, very good at managing and preventing toxicities for patients with CAR T cell therapy. There is a lot of bad press about the CAR T cell therapy toxicities, and I really do hope to tell the audience today that we are much more manned with more resources, also many more effective tools, because we now exactly know when this happens, what can be done, and in certain cases we can prevent it. As a result, a lot of times patients who are older can be absolutely safely taken to CAR T cell therapy. It's very different than assessing someone for a transplant. For a transplant, the age cutoff used to be 70, 75, not more. That is not true for CAR T cell therapy. At this point in time, I can say I have taken several octogenarians in their late 80s to CAR T cell therapy very effectively without a lot of side effects. So I do believe that this is a treatment that has to be done well, but if done well, it can actually yield excellent results. So coming back to the unique cytokine release syndrome, what does it mean? It typically manifests exactly when the CARs start expanding. And this typically happens right after the CARs get infused, and this happens over a matter of 14 days. As the CARs expand, so does the risk of developing cytokine release syndrome, which is typically high in the first week. And in the second week, patients can experience neurological toxicity, also called ICANS. Now, usually, because these are inflammatory substances, we typically give them agents that can hush the inflammation. And our biggest friends here are steroids and an agent which targets IL-6, which is an inflammatory cytokine called interleukin 6, with an agent called tocilizumab. And with that, 95% of the times we can effectively resolve patients who develop these unique side effects. Because scars affect B cells, patients can be at risk for hypogammaglobulinemia, which I'll go into detail later. But we don't quite understand why patients may also develop low white count or low hemoglobin or low platelet count. I think it is more of an immune effect, and that is something that we will closely monitor after CARs are given. Usually, the first 14 days after CARs are infused, patients are managed and observed very closely. Sometimes some centers do it on the inpatient side, meaning hospitalize the patients for 14 days. Most of the centers are now capable of doing this outpatient without any issues. After 14 days, the remaining 14 days, you're typically evaluated in a clinic setting 2 to 3 times a week. And by day 30, you actually do start noticing that the counts recover. Patients, of course, have been usually feeling well through all of this. Sometimes during the first 14 days, patients, of course, have cytokine release syndrome like toxicities. If they do, that manifests as low blood pressure, sometimes shortness of breath, but mostly fevers. It looks like as though patients may be having infection. So a lot of times patients are also covered with antibiotics just to make sure we're not missing an infection. Other CAR T cell therapy toxicities can occur. So the one thing that can occur is something called B cell depletion. It's also called an on-target off-tumor toxicity. What does that mean? So as you know, CARs affect and sit on the CD19 protein on the lymphoma cell. This is the malignant B cell, which is the lymphoma cell. Therefore, the lymphoma cell gets eradicated. But remember, normal B cells that we all live with also has this protein CD19. As a result, a lot of patients can have B cell aplasia. Now, patients can or people can live without B cells. It is not something that can lead to death. As a result, this is a safe way to eradicate tumor. But because of the B cell aplasia, remember the B cells are also where you get your antibodies from. One of the antibodies that your doctors may actually monitor is an antibody called IgG. And preemptively or prophylactically, if the IgG goes under 400, we infuse our patients with IVIG, which is basically an intravenous infusion of IgG so that you have antibodies to fight off other illnesses like infections. The infection risk is typically higher in the first 3 months than in the remaining 9 months. In the first 3 months, we typically give patients antibiotics that cover particular pneumonias like PCP. We want to make sure patients don't have any viruses, and therefore you might be put on agents such as acyclovir or valacyclovir. These are all very manageable. A rare set, I would say 30%, can have cytopenias. And the idea as to why cytopenias happen, we don't quite understand, but it may be more driven by an immune mediated effect. Therefore, usually 30% of patients may express or have cytopenias beyond the 30-day mark. And therefore, sometimes, even if mostly the patients get spaced out after 30 days to come to clinic only twice a month or once a month, for those patients who express cytopenias, we want to make sure that their counts are adequately repleted with transfusion support or growth factor support, and thus make sure that they don't have any complications from the cytopenias. I think the elephant in the room is, of course, the economic toxicity. And with regards to economic toxicity, it's absolutely important to recognize that it is a product that's nearly $400,000. Usually, insurance companies will cover it. However, the idea is to make safer tools so as to make sure that these patients can also get products that are economically sustainable for the entire system. So in terms of the next, I'm going to move forward with understanding what makes it a safe CAR T delivery. As you may imagine, not every program can do CAR T cell therapies because it takes a lot of resources. We want to make sure that the academic center or the center where CARs are done is well trained from a staff education standpoint, making sure that there is adequate oversight, making sure that there are algorithms in place, making sure we have the medications to give if patients have the unique side effects. And this is really what will encompass a safe CAR T delivery. Therefore, it's absolutely crucial to go to a center which is almost like a well-oiled machine who has done CAR T cell therapy for years. At this point in time, there are several FDA-approved constructs. I focus primarily on large B-cell lymphoma, but I want you to know that CARs are now available also for follicular lymphoma as well as mantle cell lymphoma. Now, I would spend probably just 5 more minutes on the other approved constructs for other subtypes of lymphomas. So with relapsed follicular lymphoma, it's an indolent disease. So a lot of times patients could watch and wait. Sometimes they might just get rituximab, but there is a subset of patients that will need treatment every so often. And in those patients, the outcomes are actually not as good as compared to the overall follicular lymphoma population, especially if patients relapse within 24 months of receiving initial chemotherapy. We know we want to do something more because their outcomes are compromised. As a result, Axicell was tested as a phase 2 trial, meaning a single arm study, which was done in patients who had relapsed after 2 lines of chemotherapy or 2 lines of treatments with follicular lymphoma. And the good news is that the progression-free survival, meaning the time in remission, was excellent after CAR T cell therapy. The overall survival was excellent. In fact, most of the patients are still alive, doing very well, and they are progression-free. With regards to cytokine release syndrome, this was Axicell. Remember, we talked about the CD28 product. With CD28, we do recognize that we do see cytokine release syndrome in about 80% of patients, neurological events in 60%. But I also want to make you aware that usually these are low grade, meaning high grade would be grade 3, grade 4. These would entail ICU admissions. And with Axicell, with this clinical trial, we saw about 7% of patients with high-grade cytokine release syndrome. And with neurological toxicity, we saw about 20% of patients getting neurological toxicity. Now let's talk about another clinical study using the 4-1BB costim molecule, which was tisagenlecleucel. And with that, in patients with follicular lymphoma who had relapsed after 2 lines of treatment, the results were excellent. 86% of patients responded, of which 70% had a complete response. And you can now see the difference. The high-grade cytokine release syndrome was only 0%, basically zero. Grade 3 or higher neurological events was only 1%. So clearly, 4-1BB products may have lower toxicity, but I will also say that, for example, Lysocell takes about 4 weeks to manufacture versus Axicell, which is a CD28 molecule. It takes only 17 days to manufacture. So sometimes your physician or your primary oncologist may select one product over the other only because maybe their center has more experience with the product, or they really want to get the product back for a tumor burden that is actually proliferative or growing at fast pace, and we want to keep a check. So there are many factors that go around assessing which construct to use. I'm also going to show you some data in mantle cell lymphoma. Now, clearly, mantle cell lymphoma previously, we only had chemotherapy. Then the outcomes improved when BTK inhibitors got FDA approved. BTK inhibitors like ibrutinib, acalabrutinib, zanubrutinib, it really improved outcomes in patients with mantle cell lymphoma. We also have patients that fail response to BTK inhibitors. And in those patients, previously, there were really no good outcomes. And this is where I will say Axicell made a huge splash based on, not Axicell, sorry, Brexicell. Or brexicaptogene autoleucel. It is a CD28 costim domain molecule, and it was evaluated in relapsed refractory mantle cell lymphoma patients in the ZUMA-2 study. And you can see the plateau is beginning to emerge. Nearly 67% of patients have been in a complete response, even at a longer follow-up. But this comes at the cost of slightly high-grade cytokine release syndrome, which was 15%, slightly high-grade neurological toxicity, which was at 31%. Now, Lysocell has also been tested in relapsed refractory mantle cell lymphoma with the same responses, which were very good. And now you can see there is a difference in the cytokine release syndrome, which was high grade at only 3%, and the neurological toxicity was 13%. Currently, lisocabtagene is not approved for relapsed refractory mantle cell lymphoma, but we do expect an approval later in the year, so that will be one additional tool for patients with relapsed refractory mantle cell lymphoma. So I do believe that the future of cellular therapies is overall extremely promising, but there are certain challenges that do remain. For example, we are now getting into a unique problem where patients now relapse sometimes after even getting CAR T cell therapy. About 30 to 40% of patients will not respond to CAR T. So it'll be important to understand why that happens. Sometimes the cancer cell gets smarter. It starts not recognizing the CARs attack CD19. But sometimes the cancer becomes so smart that it just doesn't express CD19. So the CARs have nowhere to bind. So maybe we can make CARs that now target two proteins and trick the cancer cell. So you may imagine the permutations and combinations that we can do as clinical researchers to make CARs more effective. The bigger issue here, of course, is the manufacturing time. I talked about the 3 to 4 week wait, where usually we may have to give bridging treatments to patients. But sometimes these bridging treatments don't work. So it's important to really make CARs that are faster with regards to manufacturing time. We also need to think about making off-the-shelf CARs, meaning instead of taking blood and making CARs from the T cells of patients, could we do that with maybe donors and make CARs that are healthier? Maybe these are off-the-shelf products where the manufacturing time just goes away. At the end of the day, we really do want to make sure that the next iteration of CARs or next iteration of cellular therapies are way more efficacious than what we have even today. But of course, we want to make sure that they are way less toxic as well. It's important to make sure that early referral happens. I think I will say, as a lymphoma doctor at an academic center who has done CARs for years now, I will say the biggest issue here is late referrals. And, I think that's where patient education is key. It's really important that even if there is very little hope that may be given, it's important to still hang on to that hope. It's important to explore other options. It's important to go to academic centers which have clinical trials. Anything we do today came out of clinical trials. We no longer do sugar pills kind of trials. That is something that's unethical. I will say that a lot of data that I presented came out of clinical trials that were open at my site. So my patients got the benefit of clinical trials nearly 5 years before it got FDA approved. It's important to really make sure that we racially include everybody. We make sure that the quality of life for our patients is preserved. But I do think that this comes from early referral. So if in doubt, I do tell my primary oncologists of my patients that if in doubt, if you don't know whether the patient is going to be CAR T eligible or not, it doesn't matter. Just send the patients for an evaluation. I will say transplant eligibility is very different from CAR T eligibility. So patients may be transplant ineligible, but that doesn't make them CAR T ineligible. There was an older patient CAR T study that has been done, and it has taken patients as older as 85 to CAR T cell therapy without undue toxicity. Of course, the next would be financial factors and make sure that we absolutely look at the cost effectiveness. We do know of studies which have been done where CAR T actually does turn out to be cost effective because if we cure patients with CAR T, the downstream costs that the patients may incur because of other treatments get negated. So, of course, we need to also make sure that we have a better reimbursement structure with regards to payers. Now, of course, there is a lot of activity going on with regards to what to do to make CARs more effective. I talked about targeting two antigens. I talked about using allogeneic CARs, which is off-the-shelf donor CARs. These are all in clinical trials. For example, I do have a clinical study which is currently targeting CD19 and 22 with the idea that maybe that may be even better than what we have right now. But it can only be done within the compliance of clinical trials because we want to make sure it's done safely. So I leave you again today with all of these immunotherapeutic agents that are available in 2023, some which are FDA approved, some which are clinical trials that are currently ongoing. Of course, the future is combining all of these cellular therapy products with the hope to cover and cure 100% of patients with lymphoma. I will say that LRF is an excellent resource. And if in doubt, pick up the phone, call the helpline. You will get the answers with regards to which clinical trial is available within where I live. How many miles away is this center? If you really are not able to look it up online, LRF is here for you to help you. We also have our annual North American LRF Forum that is happening in October in New York City. I would want to encourage most of you to join that. There are multiple breakout sessions. Today I covered CAR T cell therapy in all lymphomas, but remember, we are also going to talk about individual lymphomas at that forum. So with that in mind, I will say that a huge future awaits for us where we are hopefully going to continue to better understand how lymphomas work so that we can better target them. And after this, I'm going to leave some room for questions. I hope this was an informational session for all of you.
Hey, thank you so much, Dr. Kamdar, for that great talk. We have some questions coming in, so we'll spend the last 10 or so minutes addressing some of these. The first question is, I am a recent successful CAR T patient for DLBCL. If they relapse, would they be eligible for the race CAR trial?
So I think, like I said, the bigger, I would say, a novel issue that has now occurred is patients who may relapse after CAR T cell therapy. So I would say every academic center, every community center that does clinical trials are actually refocusing their clinical trial portfolio to include patients who have relapsed after CAR T cell therapy. So there are just so many clinical trials that are ongoing. With regards to FDA approved products, there is something called a bispecific T cell engager, which targets CD20 that has been FDA approved. It has shown responses in patients who fail CAR T cell therapy, and it's an off-the-shelf product. So, of course, besides clinical trials, that is one other option that we now have handy for patients who relapse after CAR T cell therapy.
Okay, thank you. Our next question is, can you explain the interaction between prior bendamustine treatment and success of the CAR T?
I think this is an excellent question and also an extremely important question. Bendamustine is a chemotherapy. It is an extremely well tolerated chemotherapy. People can work through Benda. You don't lose hair. There's very little side effect profile of bendamustine. As a result, bendamustine is used a lot for patients with mantle cell lymphoma, with follicular lymphoma, with marginal zone lymphoma. We typically do not use bendamustine for patients with aggressive lymphoma. But the one side effect of Benda is that it actually makes T cells less functional. Now, you may imagine if somebody is pre-treated with bendamustine, and if that patient relapses, then if I want to make CARs from that patient's T cells, and if they have been recently exposed to bendamustine, the product that I can get from that patient's T cell is not going to be as good. And therefore, based on a lot of research that has been done, we will say that typically we limit the use of Benda prior to T cell collection for at least 6 months prior. That's been our programmatic approach, and a lot of other centers as well, to make sure that our patient's T cell product is good.
Great, thank you so much. Our next question is, does presence of TP53 mutation and deletion affect the ability of CAR T cell success with mantle cell lymphoma?
Another excellent question. I think, this is a question that, of course, keeps us all awake at night because a cancer that harbors TP53, it basically makes that cancer resistant to chemotherapy. As a result, usually we right away want to give patients who express or have this mutation get agents like a BTK inhibitor, like zanubrutinib, acalabrutinib. Now, usually the responses are good, but the durability is not that good. So whenever I have a patient who has a TP53 mutation, I of course give them a BTK inhibitor to start off with, get their disease in remission, which usually does happen. But I typically don't wait for them to relapse. I take them to a CAR T cell therapy construct. On the pivotal study, which is the Zuma 2 that I showed you, about 6 patients had documented TP53 mutation. And I do know that those patients responded. On the Lysocell study, 25% of patients had TP53 mutation. They did respond. But of course, the durability is definitely something that can be improved in this subset of patients, because over the course, we have understood that these patients may not have as much of a durable response. But that doesn't mean that they will not. So I wouldn't keep CAR T cell therapy away just because you have TP53 mutation. In fact, that is my go-to therapy, but I will probably monitor that patient very closely.
Our next question is, what is the neurologic toxicity of CAR T?
So neurological toxicity, also called ICANS, can be very low grade. It could be just tremulousness to maybe just cognitive difficulty, meaning I'm just not able to say what I really want to say, versus it can be moderate. For example, it can be slurring of speech. It could end up looking like you have limited strength in your arm or in your leg. It can be pretty severe. Thankfully, that is very rare, but the severe neurological toxicity could look like complete obtundation. So the patient is unresponsive. In rare scenarios, patients can have seizures. That can happen. But like I said, mostly the low-grade toxicities are more common. But with the tools that we have, that also can be resolved.
Thank you. Someone had asked, would the exorbitant cost of CAR T cell therapy prevent it from ever being considered as frontline therapy for any type of lymphoma?
I think that is certainly something that we are exploring within research. I do believe that when we do cost effectiveness analyses, mostly we have seen that if we get our patients cured earlier, then the downstream costs that we have as a result of second line, third line, fourth line agents typically gets negated, and thus the frontline possibility of utilizing CAR, I think, is there. But at the same time, it's important to keep looking and making sure that we manufacture CARs that are not as expensive. Where is the actual cost that is driving the $500,000 worth product is, of course, a question for maybe another discussion later. But I think we are absolutely considering, and we continue to consider cost, and make sure that that also ends up being considered when we look at clinical trials. So there are many clinical trials, especially say in developing countries like China and India, where they are actually making products that are only $25,000 worth. So it's not that it cannot be done. It absolutely can be done, but we want to do it safely, so it can only be done within a clinical trial pathway.
Great, thank you. A number of people have asked, just to clarify, like for indolent lymphomas, is CAR T just not as an effective therapy?
So I actually beg to differ. I think CAR T cell therapy is very valuable in patients who have indolent lymphomas that have relapsed after multiple lines of treatment. The appeal to me for CAR T cell therapy is the fact that it's a one and done treatment. You don't have to take it for the rest of your life. If it works, it actually does work for a long time. Now, right now, we are definitely kind of crippled a little bit, for lack of a better word, because our follow up is short. But as long as we have a good follow up, like with the aggressive lymphoma CAR T trials, we have a follow up of 5 years. We know 40% of patients can be cured, which previously we could not cure those patients. So I think with indolent lymphomas, the ability is that it can actually have a better impact in my mind with the data that I showed you with follicular lymphoma, with axi-cell, with tissa-cell. These patients continue to be in a remission. And I do believe that some of the high-risk indolent lymphomas, like the early relapsing follicular lymphomas, patients who have a high tumor burden, I think these are some of the patients that, currently CAR T is approved in the third line setting. Now, clearly, bispecific T cell engagers are also approved in the third line setting. But if you ask me, I will say probably because I do have a well-oiled machine here with regards to all the resources, staffing support, and all the logistics of CAR T being taken care of at my center, my go-to is CAR T because it's a one and done treatment. And if it's done well, safely, I think patients are enjoying a remission without having to be on medications. I think that is a clear win-win for me.
Great, thank you. We have time for a few more questions. Someone asked for outpatient therapy. How do you prevent patients from getting infections at home?
I think at this point in time, the infection risk is there. I would say just being more savvy, and making sure that you hand wash, make sure you stay away from people who are sick. If your counts are low, then I will say making sure you mask up in public places. I think that would be important. But outside of that, the infection risk is not as high as it is, say, for patients who go through an auto transplant, and that has been actually data proven from all the CAR T studies that have been done, which compared them to auto transplant. So your counts may be low, but it doesn't translate into a higher infection risk. But I will say that continue to be cautious and also make sure that you look at, like, for example, the flu season. The flu season is upon us now. And if the rates are rising, then you just want to make sure you mask up, especially if you're in the first 3 months after CAR T cell therapy.
Great, thank you. A couple people had a couple questions about when you talk about cure, do you mean the cancer will never come back? And after how many months or years can you consider a CAR T patient cured?
So I will say that for us, when we take care of patients with aggressive lymphomas, usually aggressive lymphomas do not tend to come back after 5 years. And that is why the 5-year mark becomes quite important to say that with confidence. And based on the follow-up data that we have in patients who got axi-cell, lyso-cell, and tissa-cell in relapsed refractory aggressive lymphomas, I think if at the 5-year mark, 40% of patients are still in a remission, we are I would say, assured to say that the patients are more than likely to be cured, which means that that lymphoma will not come back. Now, for indolent lymphomas, it's a different story because indolent lymphomas by biology are quite different. They have a tendency to come back with the tools that we currently have. We don't have a 5-year follow-up or a 10-year follow-up with CAR T cell therapy for indolent lymphomas. So I would not rush into making that assumption so soon for patients with indolent lymphomas.
Great, thank you. And our last question is, what is the role of bispecifics in CAR T?
So, so I guess I probably would have liked to readdress that question. Are you talking about bispecific CARs or are you talking about bispecific antibody treatments? I'm assuming you're talking about the latter because bispecific T cell engagers, also called BiTEs, are now FDA approved in patients who have failed in patients who have failed 2 lines of treatment. So for the patients at this point in time, you can have CARs that are available in the third line space, as well as bispecific T cell engagers. I think bispecific T cell engagers have unique advantages of their own. They are basically off the shelf products, they're antibodies. The way they work is they have 2 prongs. One prong sits on the lymphoma cell expressing CD20, the other prong sits on the T cell and directs the T cell, which is your fighter cell, to kill the lymphoma cell. So again, very unique way of targeting cancer. But at this point in time, in terms of my algorithm, there is more data to support that bispecific T cell engagers work after CAR T cell therapy doesn't, but the reverse is yet to be established in larger cohorts of patients. So until that happens, my preference would be CARs first and then bispecifics later in the third line setting, because there is an established curative value that has been shown with CAR T, which currently, just as a function of follow-up, we don't have with bispecifics.
Great. Well, thank you so much, Dr. Kumdar, for this wonderful talk. We've received a number of positive comments in the chat already, really thanking you for this thorough talk. And for everyone who would like to rewatch this presentation, you'll be sent a recording via email in the next couple of days. So we'd just like to thank Dr. Kumdar again for joining us. And also we'd like to thank our sponsors, Genentech, Kite, a Gilead company, and Novartis for making this program possible. And as Dr. Kumdar mentioned, please remember if you have any additional questions about this or helping finding trials, you can also reach out to our helpline at 800-500-9976. And with that, I'd like to thank everyone for joining us and to have a wonderful day.
Thank you, everyone.